So a lot of the work that we just talked about was done in the sea slug, right? Aplysia. Really simple circuitry, really well-defined patterns of connection. And while I think that that was great and the mechanisms have held up to this day, there was a desire to understand plasticity in a mammalian system. And indeed, that has been done more in the last 20 or 25 years. And so what we're going to talk about next are LTP and LTD, long-term potentiation and long-term depression. Right? And so these are changes, plastic changes in mammalian circuitry. And mammalian synapse strength. Okay, and a lot of the work that is done still to this day on LTP and LTD is done in the hippocampus. So we should mention the hippocampus a little bit. I put this picture in here not because it's a great artist rendition, but I want you to see in humans, right? There are two hippocampi on either side of the brain, in either hemisphere. And we know that they play a critical role in learning and memory, right? So what we can see is that the hippocampus lights up, so to speak. So we can do fMRI studies. An fMRI detects blood flow. And so when human patients are given a learning or memory task, right, at the same time as the fMRI is being used, so fMRI is non-invasive and it's just looking at blood flow, what we see is that the hippocampus, the hippocampal blood flow, goes up. Okay. There have also been lesion studies. If the hippocampus is lesioned, the ability to form new memories is lost. There's this famous patient, HM, and so I think one of you is maybe a psychology major as well. And so HM, this patient had damage to his hippocampus, and he couldn't remember what he did the day before. He couldn't remember what he had for breakfast, but he remembered things that would happen 20 years ago, right? And so when he died, his brain was sectioned and sent off to all of these labs to study hippocampal function, okay? Another sort of line of evidence that the hippocampus is required in learning and memory tasks is that there are individual cells within the hippocampus called place cells that have been extensively studied. And these place cells only fire action potentials when in certain location. So people will talk about spatial memory, right? And we think that spatial memory is encoded in the hippocampus because of these cells that we see only firing their action potentials when in a particular spot. So if you're going to look for long-term plasticity, I think a good place to look would be in an area of the brain that's going to store memory or be involved with learning new information, because we know something is going to have to change in those synapses, right? So these are three, I think, pretty good lines of evidence that the hippocampus is a good place to look. All right, we need to talk a little bit about what we'll call I've used this term before, maybe the cytoarchitecture, that is how the cells are arranged within the hippocampus, okay? And so I think maybe we can look at this picture here first. What you see in blue, right, that's all of the cell bodies. Basically, the blue stains the nuclei of the cell bodies. And so you see that the hippocampus, what they've done is they've cut it in cross-section, right? So you've got the hippocampus seen in this picture. You could imagine cutting it in cross-section. Okay, so if we cut it through here, this is what it looks like in cross-section. And we see what look to be almost like two different C's that oppose each other. So there's one here and one here, right? You could say that, that's loosely analogous to this C here and this C here. Okay. These are layers of cell bodies, okay, where they have nuclei. And so there's not cell bodies throughout the hippocampus. There aren't, instead, all of the red that you see stained are axons. Okay, so, if we pick an arbitrary point here, right, there's very few nuclei out here, right? The nuclei are in these layers where we see a lot more blue staining, right? So this would all be cell bodies and nuclei. And we can subdivide the hippocampus into different regions, right? And so here's the CA3 region, which contains a lot of what we call CA3 pyramidal cells. And these pyramidal neurons, right, send out axons that make synapses with neurons that have their cell bodies in the CA1 region here, okay? So let's put some of this up here. So we can say that the hippocampus resembles two opposing C's. Right? And where we have those letter C's are where we see cell bodies. So the cell bodies are densely packed, whereas the axons make up sort of the rest of the hippocampus. Okay, so we're going to talk about one particular connection between the CA3 and the CA1. Okay, so there are regions within the hippocampus, subdivisions based on what we know about the circuitry. Right, there's a CA1 region and a CA3 region, and these are going to be the most important for us. The CA stands for, it's Latin for a phrase meaning Ammon's horn because of the way that this structure looks. And the idea here is that cell bodies that emanate from CA1, so maybe let's go over here. Cell bodies in CA1 have axons that project, so send out axons, that synapse in the CA1 area. So bodies in CA3, I should say. Send out axons to CA1 dendrites. Okay, so that's what's shown here. Right, here is the CA3 pyramidal cell that we're going to focus on. Its cell body, right, is in this dense layer. And its axon makes a synapse not in this cell body layer, of course, but its axon actually projects to an area where the axon terminal connects with the dendrite of CA1 neuron here in between these layers, right? And so there's not a lot of cell bodies in here. This is a lot of synaptic connections, axons and dendrites in this area, okay? So we're going to focus on this synapse, the synapse between CA3, what are called pyramidal neurons, and CA1 pyramidal neurons, specifically the axon terminals of CA3 pyramidal neurons to the dendrites of CA1 pyramidal neurons, okay? We may come back to more of this architecture later, but I want you to have some sense of that, right? So I think that's good enough for now. This pathway that we're going to talk about from CA3 to CA1 is so well studied it actually gets a name. It's called the Schaefer collateral pathway. The Schaefer collateral pathway, okay? And like we said, it's CA3 neurons. Right, so the cell bodies of these neurons are out in CA3, and they synapse on the dendrites of CA1 neurons. So these neurons are called pyramidal neurons because they look like pyramids. But this is the Schaefer collateral pathway, so you go from CA3 to CA1. And what we see right off the bat is that high frequency stimulation right, so this is when we tetanize one of the neurons, right? When we give the CA3 neuron a tetanus, leads to LTP in the CA1 neuron. Well, let me show you what that looks like. When I looked over at this figure to show you this induction of LTP from CA3 to CA1, I realized that this is not the right figure, right? This is going to show us a similar concept. So I think what I'm going to do is wrap this video here, put in the figure that I forgot to put in, and we'll talk about where we see LTP, and we'll talk about some of the conditions that are necessary for LTP in this Schaefer collateral synapse.